not too, you can have R console than the straight R console. I like to use the straight R console because it's just a preference. That's exactly what it is for me. I don't, it's just a preference because it depends on how many screens you want to be looking at while you're getting your analysis. So let's just start. Now, maybe I should put this, does everybody have the R biostatistics codes? Or do you need me to put it inside the chat? Chat, please. Gotcha. Yes, I got it. I just uploaded it. You got a problem downloading it, let me know. All right, let's just start. So in descriptive statistics, what we're going to do is it's really like a plugging in, but at the same time, you get to understand what you're doing. So if I do in the first heading, I'm defining X1, you have R norm. R norm stands for a random normal variate. I'm just defining it as that. So once I hit enter, then I bring up X1 again. Actually, I'm sorry, I told you that wrong. It's a built-in data set inside R. That's what we're using today. So you have all these X1s. If you found all this data, you're working in a wet lab or even computational lab, to do this one by one by hand is going to be ridiculous. So let's say I wanted to find the mean of it. Did that single line work for everybody, the mean? I mean, if it worked for you, great. If it didn't, you can just ask me a question as I go through, because since I'm sharing the screen, it's kind of hard for me to look at the chat box. So just, it's an open forum, so just let me know. I found the mean. What's the median? You got two values to give you, the mean and the median. They're pretty, not too far from each other. They're actually kind of close. So what would that tell you about your data set is like? You need to ask that yourself, given what we learned about the mean and the median recently. Now, if I want to calculate its 25th and 75th percentiles, so you use the quantile function in R. So the quantile X1, which is the data set, 25th percent, 75th percent. You have to change it to decimals. The C is just means it's a number. 25th is this number, 75th percent is this number. Now remember, you used I believe it's the Q3 minus the Q1 to get the Q2. So at 50%, you can figure that out. the minimum value, x1. And the maximum. You got 996 lines with 4 rows. So that's 990, that's almost close to 4,000 numbers. If you can see where the min is and the max is just by looking at it, you have very good eyesight. But the problem sometimes I run into explaining this to students is that they don't like coding, they don't like scripting, they don't like too much dealing with computers. But the computers is here to make your life easy. The big thing is work smarter, not harder. So we're going to do what they call summary of all this X1 data set. Mean, the minimum, first quartile, median, mean, third quartile, and the max. Any questions what I've done so far? Just speak up verbally. If not, I'll keep going to t-tests. Okay. Now, the next example, what we're doing is daily energy intake in kilojoules for 10 individuals. We're going to define our data set. Energy intake is the name of the data set given by these numbers. All right. And to make sure it works, all we do is type in energy take. Should give you those numbers back, no problem. We're just giving all these numbers a name. I say, now let's find the mean of the energy intake. Energy take, I should say. That's your mean, which is right between these two numbers. Now we're going to see if the energy intake deviates from recommended 7,600 kilojoules. The mu is what we're comparing it against. We're doing a t-test, of course. Now, from the data energy take, your t statistic with the 9 degrees of freedom, your p-value is less than 0.05. So if I ask you, which one did we prove? Did we prove the null hypothesis or did we prove the alternate hypothesis? You can answer, whoever knows, you can answer me. I'll let you think about it. Let's move on. The next thing we're going to be covering is a two-set. Mr. Davis, explain yourself.
Well, my logic, well, my rationale behind it was if we were, if the p-value has to be over 0.0, I think it's 0.05, and our current p-value is less than, so we would accept it, right?
Well, the p-value has to be less than 0.05, right?
Right.
Now, if the p-value is less than 0.05, that means you're finding a difference, correct?
Correct.
If you're finding a difference, would that tell you that the null is correct or the alternate is correct?
Ultimate.
There you go.
Okay, sorry, I worded that wrong. My apologies.
That's no, don't worry about it. We're all here to learn. No worries about it. I'm not even going to tell Dr. Wu on you.
Appreciate it.
All right, we're going to a 2-sample t-test. Before we do that, we have to install this package, ISWR. Crane mirror. So how R operates. Technically, you can do pick any one of these you want. I'm more comfortable with the Ohio side in the United States. Now the package was downloaded. Everything successful is unpacked. Now we're gonna load it. Say unpack it, then you load it. When you're doing R scripting, make sure, please pay attention to where the period goes and where the quotation marks goes with the parentheses. Now, if you need me to zoom in more on this, I can, but I think you can see it well enough. To make sure the library is installed correctly. It's in there, so we can keep going. I'm using an older version of R. I need to upgrade to the 4.1, but I'm at the 4.0 right now. Now, we're going to attach energy intake. This is the data on pre and post-menstrual energy intake in a group of women. And we're going to make sure intake has been successfully loaded. So pre versus post. That's your data set. You got 11 samples of each. So we're going to do the post minus the pre. Obviously, you're going to see negative because the post, actually, no. The post is, yeah, I was saying the correct. Post is relatively negative compared to the pre. And you do a t-test. Mr. Davis, vindication time. Any reason I'm picking on you is because you were a student in biostats too.
Oh, I wasn't one of your students.
Okay, I'll still pick on you.
That's what's up.
Tell me, given that p-value.
It'd be the same as the previous problem that we worked out because there's several zeros in front of that p-value.
Now, given this p-value here, what can you infer from the null or the alternative hypothesis?
That there's a true difference in the mean, there's a difference between the mean.
All right. Now, what does that tell you about the hypothesis you're testing? Remember, we got pre-menopause and post-menopause, right? We're measuring energy. So if you get a very low p-value compared to the tested p-value of 0.05, which hypothesis is correct? Is your null or the alternative?
This one would still be the alternative.
Yeah, the alternative means the energy pre does not equal the energy post. Given the numbers, you can find that, but this is just a simple data set compared to what you might experience in your research careers. Now we're going to move into something called independent samples, where one sample doesn't depend on the other. So what we're going to do again is go back to the energy data set. We're going to attach energy. Make sure it's in there. Now you need to, as I said before, I'm gonna go back for something. You're given these data. Make sure you know your hypothesis. You have to know your hypothesis, the null, excuse me, the null versus the alternative. You have to set that. Null hypothesis, the means of both columns are the same. Alternative hypotheses, there is a difference. I'm just saying that shorthand, but you have to say it, like there is a difference in energy expenditure between premenopause versus postmenopause. That's your alternative. Or the null is both of them are equal. So make sure you know remember your hypothesis, because you're going to get all these numbers and you might not know what to do. All of these is just helping you getting a conclusion of the data after analyzing it. So I'm going to do a t-test. The next line of code, as it says in the work along, we're testing whether there's a shift to the level between the two groups. P-value is very low. I'm not going to ask Mr. Davis anymore. I'm not going to ask anybody else, but you can infer from that p-value which hypothesis is correct. And if you don't understand, let me know. I'm glad to go over it again. Now remember I said we're gonna do a box plot comparing the two variables. So all I'm doing is plotting expenditure and stature. Expenditure being this column. Stature is if you're lean or obese. Voila. Lean has some outliers, which can be the outliers are like 11. Yeah. And low, it's pretty low too, because essentially it's supposed to be between 7 and maybe 8.5 versus obese. Now, when you see that kind of graph, sorry, remember this, you're going to see a difference. Because if you didn't see a difference, they'll be on par. Like this line would be equal to that line, but you see there's a big difference. Now we're moving into analysis of variance, which is one and ANOVA. We're going to do a one-way ANOVA. Which is, we're going to be using the ISWR package again. It's already loaded, but I just want to make sure there's no problems with it, so I'll just test it again. If you ever wanted to look at the, I think I showed this before, but there's specific columns you can look at inside the data set. We're going to attach the one called red cell foliate. And we're going to read it. To look at it, red cells versus ventilation. This is a built-in data set inside R for use for examples. Again. Now let's just run a summary. Minimum, first quartile, median, mean, third, max. Notice in the ventilation data set, the ventilation column, you can't really do a mean, third quartile, and all that stuff, the max with these kind of numbers. These values, because they're not numbers. Let's do an ANOVA, one-way ANOVA. Now remember, in the one-way ANOVA, to have a big difference, this number has to be very small. So really, we're at 0.05. Now, if you can recall what the p-value is for the ANOVA, it will help you explain your data. Like, if you're given foliate versus ventilation, what will the F statistic that you get, that you calculate, say about in relationship to the p-value? You can answer that yourself. I'm not going to ask anybody. Any questions? If I went too fast, let me know. I'll be happy to go over some things.